Welcome to part 2 of the video. So here we're taking a look at some implications of osmosis. So if you look at the figure, we see essentially three situations. On the far left-hand side, the top left figure shows us a bacterial cell in an isotonic solution. So essentially, the amount of solutes in the water is the same, both inside the cell and then outside in the solution. That is showing us a bacterial cell, or one, and that's what it is, a bacterial cell, but essentially it has that cell wall. If you take a look at the cell below, this is a cell without a cell wall, so it's showing you the same thing. The cell's just happy, and net movement of water is zero because it's equalized or at equilibrium. Okay, the center figure, hypotonic solutions. So the solution we're referring to is the solution that's bathing this bacterial cell. And yes, bathing the bacterial cell. So if you take a bacterial cell, and we know that it has a normal concentration of solutes inside the cell, and now we put it into a solution that has less solutes and more water, we already know what water wants to do. Water wants to move from a region of high concentration to low concentration. So you're going to get the net movement of water moving from outside the cell inside the cell. Now, so the cell can essentially take up that water and bloat. Because it has a cell wall, it's not going to burst. But take a look at the cell without the cell wall in the figure below. If this is like an animal cell, what's going to happen is our cells, when they take up too much water, they're going to explode. So essentially, that's due to the differences in tonicity. So cells being put in a hypotonic solution. The far right-hand side shows us what will happen if you take cells and you put them in a hypertonic solution. So a hypertonic solution has more solutes and thereby less water compared to inside the cell. Water is going to move from high regions of concentration to low regions of concentration. So in that hypertonic solution, there's going to be a lower concentration of water because there's a higher concentration of solutes. So the net movement of water is out, and this causes that bacterial cell to shrink. What happens in the animal cells, like we've done this with blood cells, experiments with blood cells, and essentially they shrink. So and they might lyse, which means they just split apart or they just, disintegrate. All right, moving on. Okay, so cardinal temperatures. So essentially, cardinal temperatures tell us the maximum, minimum, and optimum temperature that microorganisms can grow. So remember how I said this is super important earlier in part 1 lecture? So essentially, if for example, food microorganisms, if we know that a particular food microorganism has a minimum temperature, and that minimum temperature can be controlled by us setting our refrigerators at a certain temperature, then we can essentially help reduce or even eliminate the growth of that bacterium in our food. So it just is part of thinking about that practice of food storage and preservation and making sure we don't have food spoilage. Okay, so the minimum temperature tells us the lowest temperature that permits a microbe's continued growth and metabolism. So it can still, grow and metabolize and do its thing, but below this temperature, its activities stop. The maximum temperature is then the highest temperature at which growth and metabolism can proceed before how proteins are denatured. So if we heat up proteins, they're going to be denatured, which means then we've lost the ability of some of those enzymes to be doing their critical job. Lastly, the optimum temperature is the intermediate between the minimum and maximum, and it promotes the fastest rate of growth and metabolism. Generally speaking, it's not just one number, but it's a small range, like essentially their optimum temperature. I laugh and say my optimum temperature is between like 67 and 71 degrees Fahrenheit. That's just my optimum temperature. I get hot above that and I get cold below that. So something to think about. So when we take a look at bacterial bacteria in nature, we can start to see that think about all the different kinds of biomes or types of climates on the earth. Everywhere you can imagine, there is a niche that bacteria can grow or other microbes can grow. They're going to grow. So even the hottest places on the planet, these extreme thermophiles here. So they are living between like 67 degrees Celsius up to maybe 132. Okay. Down here on the other extreme, we have sacrophiles, which really like the cold temperature. And so they can range from negative 20 to 15 degrees Celsius, but there's that optimum. So take a look at each of these where this is showing this little plateau sort of thing, or where this curve kind of maximizes and slopes. That is the optimum temperature. So let's take a look at some of these different ecological groups of bacteria. So sacrophiles. They like it cold, optimum temperature below 15 degrees C, capable of growing at 0. And we're going to see that once it gets too much above 15 degrees Celsius, not really going to like it. So by 20 degrees Celsius, they are not thriving. So storage at refrigerator temperature causes them to grow rather than inhibiting them. So think about this. We've all been told, oh, you need to refrigerate that. But for some sacrophiles, they're going to be they will continue to grow in the refrigerator. I did see in your book, had some really good examples. So one of those, do we have one of those in here? No, it's talking about bacteria, fungi, and algae that are growing in cold lakes and rivers and snowfields, polar ice, and in the cold, deep ocean. So rarely are these pathogenic. Okay, mesophils. So essentially tells us these are the majority of medically significant organisms because body temperature is in this range. So body temperature is 37 degrees Celsius. So we see these things can grow at intermediate temperatures between 20 and 40, but that optimal temperature for them is 30 degrees to 40 degrees Celsius. So we're thinking temperate, subtropical, and tropical environments, animals and plants. They inhabit animals and plants, both in soil and water. So thermophiles, they really like it hot, 45 degrees Celsius. And now we're thinking about the hotter places on the planet. Lava, hot lava, the bottom of the ocean floor where we have these vents. That'd be a good place for these things to grow. And then really, really, the center part of compost piles gets really warm. The thing is that most eukaryotic organisms cannot survive above 60, but extreme thermophiles can grow between 80 and 121. Let's go back 1 minute. So mesophiles, I did want to tell you about one of them. Staphylococcus aureus grows optimally at body temperature, but are also facultatively psychotolerant, meaning they can survive and multiply slowly at refrigerator temperatures. So while we think, stick in the refrigerator, that'll take care of it, Staph aureus can continue to grow in the refrigerator. We also have Listeria monocytogenes, a human pathogen. It's a truly psychotolerant organism, meaning it likes that growth between 30 and 40 degrees Celsius, but it grows slowly at temperatures as low as 1 degree Celsius and can actually grow in ice cream and refrigerated meat. So this is why it's important to know what is that intermediate temperature and the maximum and minimum of the cardinal temperatures. All right, let's take a look at gases, because we talked about earlier in this lecture, there's some serious byproducts of oxygen use in organisms. So when we think about the gases, the two big gases that affect microbial growth are oxygen and carbon dioxide. So oxygen having the greatest impact. And of course, we know it is a respiratory gas and is important in that electron transport chain and is a powerful oxidizing agent. But the thing is we've got to figure out what we're going to do if we can't detoxify it. So I told you earlier, the microbes fall into three groups. Those that use the oxygen and detoxify. They neither use it nor detoxify. And the last group is they do not use oxygen, but they can detoxify it. So this is going to become important because we will see that they likely will have a variety of enzymes and cellular reactions to handle those byproducts. All right, so if we have a single oxygen, we call it singlet oxygen. It's an extremely reactive molecule that damages and destroys cells by the oxidation of membrane lipids. So we got to figure out what to do about that. Then we have the superoxide ion, also highly reactive. We have hydrogen peroxide, which is toxic to cells, and we know that it's toxic to cells because we can use it as a disinfectant. And then we also have the hydroxyl radical. So let's see what we're going to do with these things. So we see that most cells have developed enzymes that essentially scavenge and neutralize reactive oxygen byproducts. And we see that there's a two-step process and it requires two enzymes. So remember when you see the suffix ase, you're going to think enzyme. So we see two enzymes here, the superoxide dismutase and catalase. So in step one, we see these superoxide ions and hydrogen protons that are essentially going to be catalyzed by this enzyme to make hydrogen peroxide and oxygen. So then we take the hydrogen peroxide, which is very toxic to cells, and we convert the hydrogen peroxide to water and oxygen. And that O2 has all of its electrons freely paired, so it's O2. And in this form, it is not reactive. So catalase is going to break down hydrogen peroxide to water and oxygen. Okay, so let's go ahead and take a look at oxygen tolerances of our microorganisms. So as you might imagine, some microbes really need lots of oxygen, and they require it, and that's where they're going to grow. They're not going to grow without oxygen. So take a look at the test tube on the right-hand side at the top of this figure, and you're going to see that yellow kind of halo at the top of that test tube is showing us the bacterial growth. So these are what we call the aerobes. They absolutely have to have gaseous oxygen. And guess what? They have the enzymes that they need to process all those toxic oxygen products. So we will call this organism an obligate aerobe, an organism that cannot grow without oxygen. So that's most fungi, protozoa, many bacteria, and of the bacteria includes bacillus species, as well as Mycobacterium tuberculosis, the causative agent of TB. So next we have the microaerophiles. So take a look at the test tube on the right. You're going to see that halo is not really right at the top. It's just below the surface. So microaerophiles are harmed by normal atmospheric concentrations of oxygen, but require a small amount for metabolism. So they live in those areas where they're not going to get exposed to super high amounts. So the surfaces of water, for example, where it meets the air, that would be an area where there'd be high oxygen. So essentially we see that these organisms live in the soil or water or in mammalian hosts, but not directly exposed to the atmosphere. So this includes Helicobacter pylori, which is the causative agent for stomach ulcers. All right, let's take a look at the anaerobes and the aerotolerant anaerobes. So anaerobes lack the metabolic enzyme systems for using oxygen in respiration. So our obligate anaerobes also lack the enzymes for processing the toxic oxygen and die in its presence. So these bacteria live in really low oxygen environments. This would include many oral bacteria and intestinal bacteria. Lower right-hand side, our aerotolerant anaerobes do not utilize oxygen, but can survive and grow in a limited extent in its presence. So they're not harmed, basically because they have ways to break down those oxygen react.